I was invited to talk today along with Max, and it's a real great opportunity to update you on some of the things that are happening here in the city. Hopefully the clicker will work for the next one. Yes. So the part that I thought I could tell you a little bit about is the title of our presentation, the joint presentation, was "Are we there yet?" when it comes to predicting and modeling Parkinson's, because you know it's an incurable disease today still, and we don't have perfect ways to halt it or to stop it, and also we don't have the perfect models to test new drugs. So we're constantly trying to do better, and you will hear from Max in a moment as to how he is modeling the illness. But what I briefly wanted to talk to you about is answering or addressing two questions. One is, can we tell who has Parkinson's disease without actually examining a patient? And that sounds maybe counterintuitive, because you know a lot of patients that have symptoms hopefully have access to a neurologist and are being seen, and then based on a clinical examination, many of which of you have seen that, the decision or the conclusion is being made, yes, you have, or early stages, or you have symptoms of Parkinson's disease. But could we possibly make the diagnosis the way we make the diagnosis of a heart attack today? That is actually just doing a blood test, and we don't even need an EKG anymore. So if we could identify who has Parkinson's, how would that help treatment in the future? And number two, can we predict illness in those people who are perfectly healthy and don't have Parkinson's yet? Can we predict who may develop it over the subsequent years? Because this is the course of Parkinson's in a graph that demonstrates the journey of 20 plus years from very early risk factors and symptoms before people develop motor problems such as tremor, slowness, stiffness. And then you can see that many patients run into other problems too, such as with bladder function or with their memory. And so wouldn't it be nice if we can actually capture people really early, the way we try to prevent today heart attacks and strokes, because we identify risk factors and act on them. And that would be really the best case scenario to prevent Parkinson's in the first place. So in other words, can we address two issues? If this is a group of people, subjects, can we pick out who has Parkinson's disease? And the second question would be, who will develop Parkinson's disease in the future? And so I often use this slide because it's inspired by one of my patients who has a genetic risk factor for Parkinson's, and what his sister said to me about 2 years ago when she came, because she accompanies him. He lives in the larger Ottawa area, and gave permission to use his photograph. And that is, Sandra, his sister, said, "It's frustrating for all of us. This disease seems poorly understood," and there's a technical term, idiopathic Parkinson's. So poorly understood, is unpredictable, and is unstoppable. And I want to inject some optimism here, because I think this is changing slowly but surely. And the cause of Parkinson's disease, you know, what Sandra was referring to, that it's poorly understood. Well, we understand more and more that there are different types of Parkinson's, such as those that start really early, young onset Parkinson's, usually before the age of 40, and then others who start after the age of usually 55. And this is a journey through life. If you follow me for a moment, this is from birth to 100 years of age of any one of us. And if you move upwards, you have higher and higher risk of Parkinson's until you cross a threshold where all the criteria are being met and you're being diagnosed with Parkinson's. So you'll see very few people in the general population get Parkinson's, and at the end of life, when people are in the 90s, it becomes more and more difficult to differentiate who is healthy versus who has mild features of Parkinson's. But there are some people that obviously have a much accelerated phase, and they go through a preclinical phase that's normal. Then they have early symptoms of Parkinson's until they finally develop this full movement disorder syndrome by crossing this threshold. And what we have come up with is a theoretical concept first. Can we quantify where each one of us stands vis-a-vis the risk of developing Parkinson's disease in the future? And the components that explain who gets high blood pressure, who gets a coronary attack, a heart attack, who gets stroke, who develops breast cancer. Many, many human diseases can be reduced to 5 factors that interact to bring out this illness. And the same is true for Parkinson's disease. There are environmental factors. There's DNA, so there's risk factors in your genes. There's I, which is, if I may simplify here, says this the initiation of tissue changes, whether it's the liver, whether it's the heart, or whether it's the brain. And it's modified by what gender you are, what sex, male or female, and the factor of time. And this explains why some people develop their Parkinson's very early, others later, or some people don't at all. It's the interaction between these two factors. And so we had this idea whether we can possibly calculate that. And this is done for when you go to the doctor and have your blood drawn to measure your cholesterol. Those numbers in your lipid profile can help calculate what your risk is to have a heart attack down the road. And the same, we have prediction mechanisms for who will develop stroke. So in these 5 factors, E, D, I, G, T, factor E, for instance, stands for environmental exposure. So people who have had, for instance, pesticide exposure, we think also certain infections, and even constipation may be grouped there because the bugs in your system live much longer inside of you if you're constipated, severely constipated already in your 30s or 40s. Then the issue of sex or gender. Men are much more likely to develop Parkinson's than women. Next one is the DNA, so the genetic risk that you have inherited from your mother and or father to develop Parkinson's disease, which may mean that you develop Parkinson's disease early in life versus somebody else just late in life. And it's not just one genetic risk factor, but it may be a combination of multiple. And it's taken together. The idea is, can we quantify this to calculate the risk? And in this gentleman, for instance, he had multiple family members with Parkinson's and another disease that's related to Parkinson's. So in him, not only do we know that he has a particular genetic risk, we can calculate it, and we can then also use this information to get him closer to clinical trials, as you'll see in a few minutes. So the simple math formula that we came up with is to just adding these all. So the Parkinson's risk calculation combines something for the environment, something for DNA, something for tissue changes, and multiply this with sex and time. And so the reason why this is important is because we could identify, is somebody who's healthy on a trajectory to remain healthy, or does something change that puts him or her at higher risk, or is somebody already advanced with his or her risk to really get closer and closer to Parkinson's disease. And these different stages are the best ones to try to do something. Why? Because as you saw in this gentleman earlier, when you have a tremor, when you have slowness, and you already have Parkinson's disease, it's much, much harder to fix it as compared to preventing it. So on this journey, what we are envisioning in the future, that what's the holy grail about Parkinson's is, can we prevent that somebody moves up here, and can we push this way? Can we protect a person's residual cells if they've already reached that threshold? And can we even restore some cells that have already been lost? So to pursue this, we have come up, we have a team that works together closely, and Dr. Xuan Li, she's in the audience. Here, you will recognize her, and afterwards when you talk to people. She's a methodologist who is an expert in prediction modeling, and we have 3 people that have made a name for themselves in helping to predict outcomes of human diseases. And so we work together very closely, and using, again, the different cohorts that we have, patients that have participated and their loved ones, to try to answer the question, can we pick out who already has Parkinson's disease when we just use these risk factors without even examining a patient. And there's 2 large cohorts that we are using with multiples of hundreds of people. The Progressive Parkinson's Marker Initiative by the Michael J. Fox Foundation is the first one, and the second one is a cohort that we've used that's called New Onset Parkinson's Disease. That's a German cohort. And what you see here is there's variables that you identify. Like, for instance, did you have a head trauma? Did you have constipation? Does that person have a reduced sense of smell? Is there family history in your family of Parkinson's disease? And then particular values that we look at that would indicate some disease process has already started. And I just want to give you 2 examples quickly where this stands. And this is what Swan has come up with in the last few weeks. In the Michael J. Fox cohort, for instance, predicting who has Parkinson's versus who is a healthy person. If you take just random chance, it would be 50% chance. But the information that Swan has put together in this formula, in this algorithm, separated in this example of over 300 people here with an accuracy of 83, 84%. So this blue line, the more it reaches up to the number 1 here, the more it gets closer to 100%. So 50% would be chance, and we're well above 50%, 83, 87% correct in predicting who has Parkinson's, who doesn't, without having examined these patients. And that would be really helpful if that can be shown to be true. So to test this, she then examined this other cohort, and Swan has shown here again 50% chance prediction who has Parkinson's, who doesn't, and this cohort is about 74%. So the closer to 1, the more we are right to predict who has the disease and who doesn't. And these are some of the factors that go into calculating that risk. So this is quite remarkable because most doctors need to examine a patient to say, does he or she have Parkinson's, and not just numbers based on filling out a form. And where this goes is that we need to test this much better. So the first step to prove whether such a formula could work, who already has Parkinson's, is to what's called to discriminate, to look for discrimination or diagnostic accuracy. So we'll look in a total of 4 cohorts at least to see, can we really pick out who's healthy and who has disease? If that's completed, or when that's completed, we also want to go back to the question, who in this group of people will develop Parkinson's in a prospective manner? In other words, everybody, let's assume everybody's healthy, and if we follow them for 3, 5, 6, 7 years, who will then have Parkinson's based on the formula and based on the variables that we have? And that's called calibration effort. And so going back in the very last slide, I think this is to try to understand how does that inform treatment. So if we're correct, and within the development of Parkinson's disease, you have 5 factors. In some people you have a bigger genetic risk, and in other people you have less of a genetic risk. In some people you have more of an environmental risk, in others less. In this gentleman, as I told you, he has a very high genetic risk. And I'm proud to tell you that worldwide, but definitely Ottawa is now part of this and has played a key role. This gentleman has a genetic risk that led to the development of a drug. And currently, as we speak, there's the first international trial in North America and in Europe that is using a drug that was developed specifically for his genetic risk to try to treat his risk and diminish the progression of Parkinson's. And this is called the MOVES PD Parkinson trial. And this gentleman has a mutation in a gene, so it was the mutation of his illness in this patient matched with a new drug, and this is a placebo-controlled trial, so it's a dummy-controlled trial. So this is the first time that this happens, and Ottawa played a role in the development of it. And so in the future, and there's about 8 to 10% of people that have this mutation among all people with Parkinson's disease. So to answer the question, are we there yet? The question is answered if can we tell who has Parkinson's without examining a person? We're almost there. I think within a year we should know whether this holds up, which would be a first, and that would be really wonderful. Can we predict illness? Who will develop it in the future? That will probably take another 2 or 3 years to answer, because we have to follow people long enough. And then thirdly, does genetic risk for Parkinson's make a difference in the treatment? And the answer is yes, we're there. The first trial has started, and many more will come. And sadly, in neurology and in medicine in general, many trials have to be carried out before the one nugget is being found that really makes a difference. But we're committed around the world in the Parkinson's research arena to push for more trials that try to identify the risk factors and match a drug for the risk. And so, looking into a crystal ball, we don't know the outcome of how many trials it will take, but certainly with the support of our patients, their family members, and the community, we're in a really good position here in Ottawa to push for that as hard as we can. So then I'll stop here. Thank you for your attention. We thought that in the interest of time and organization, we'll probably do the second presentation. So if you have burning questions, write them down, remember them, and then we'll have time afterwards to answer questions for Max and myself, okay? So without further ado, I'd like to introduce Max Rousseau. Dr. Max Rousseau was a trainee here in Ottawa. He was outstanding as a student. He then went for further training to Texas, Baylor College, and worked with a fantastic team, and in no time made really exciting contributions. He was way more productive than any one of us investigators have been when we were at that stage. So we were excited to hear that he wanted to come back to Ottawa, and he arrived 8 months ago, 7 months ago. He's had a baby since then. He bought a house. He got his research off the ground. His trainee just won a fellowship. So this man is flying. So welcome back, Max.
Thank you, Michael. That's a tall order to follow after that great presentation. So I've been tasked to speak to you about the recent advances we've made in Parkinson's modeling. And I apologize if the slide is a bit compressed. So, as Michael mentioned, Parkinson's disease is incredibly heterogeneous, and a lot of the clinical trials that are currently underway really deal with, snapshots of the disease, and they take a long amount of time. And so what we're tasked with as basic scientists is to really model the disease in other organisms to really get insight into the disease. And so why would we want to model Parkinson's disease? Might be obvious to some people, but really to treat a disease, you have to first understand it. And I hope you grasped from the previous presentation, we don't entirely understand the disease yet. We're getting better at predicting it, we're getting better at understanding its progression, but we're not there completely. And so if you can recreate Parkinson's disease in a mouse or in another model organism, in a monkey, in a fruit fly, you can better devise drug treatments and then feed that back in, obviously, to the patient population. And then learn with this cyclical experience how to develop better and better models. And so when you're thinking about modeling Parkinson's disease, where do you start? Well, as basic scientists, we have a really nice array of model organisms that we can play around with. And that may range from the budding yeast and worms, but also rodents like mice and rats, as well as non-human primates. And since we can't perform experimentation on humans, we kind of have to take a couple of steps back to make sure that we can do manipulations, try to recreate Parkinson's disease, and then bring it back up the ladder so that we can eventually treat humans. And so if we look at this, this kind of single direction of evolution, you see that these lower organisms actually are very easy to manipulate for us as biologists. They're very cheap to use. However, they're less and less clinically relevant. If I told you a yeast had Parkinson's disease, I doubt you would believe me. And so we want to make sure that our models are the best mix of clinically relevant, but that we can still manipulate so that we can recreate the disease in our own hands and test treatments on them. So while many of these models have benefits, our lab focuses on rodents, specifically mice, because mice have very similar DNA to us. They have motor movements, unlike yeast again. But they also have many manipulations that we can do to them in order to recreate the disease. And so when you look at mouse models of Parkinson's disease, what I thought I'd do today is actually give you an oversight of a couple of the models that are becoming more and more widely used or are new and interesting. So not necessarily talking only about my work, although I will have a shameless plug at the end. And so the different types of mouse models that are used are often using chemicals or toxins, as well as viruses. So you can actually use viruses to deliver genetic payloads into mice and recreate certain symptoms of Parkinson's disease. You can actually inject toxic proteins directly into mouse brains, and they develop certain features of Parkinson's disease. And then lastly, we use the genetic information that we have from humans, such that we learned in the previous presentation, to reintroduce that into the mouse to see if we can see similarities between both organisms. And so I created a word cloud here on the right to really highlight some of the various aspects of mouse models. And if you do a survey of the literature, there's many popular genes, but probably one of the most popular is alpha-synuclein. And so why alpha-synuclein? Several of you who are familiar with Parkinson's disease will know the name or the word alpha-synuclein, and that's probably for two reasons. One is pathology, and I'll explain that in a second, and the second is genetics. And so in Parkinson's disease, individuals with Parkinson's disease accumulate alpha-synuclein, and it's this protein that's very sticky and gunks up in the brain, and it's thought to damage the brain. And pathologically, we can actually see this in the brain as a form of these Levy bodies. And in terms of genetics, alpha-synuclein is also exciting because individuals, well, exciting might not be the best word, individuals that have mutations in alpha-synuclein, either mutations that increase its stickiness or increase its overabundance in the brain, can actually cause Parkinson's disease in and of itself. So there's basically two converging features that make alpha-synuclein front row and center the face of the disease. And so can we leverage alpha-synuclein to model Parkinson's disease? So I'm going to talk about 3 mouse models just in 3 slides to give you a brief overview to really get the point across that we're getting there, but we're not there yet, in the sense that we're being able to model certain aspects of the disease in these mice, but we really don't have a unified model yet, and that's what we're actively trying to push forward. So the first model will be one called the PFF model, a second model, what I'll call the 3K model, and then last our shameless plug for the alpha-synuclein NLS model, for which Haley from my lab was recently funded by you guys to study. And so the PFF model raised a big splash probably about 5 years ago, and it's built on this theory that alpha-synuclein can propagate in a prion-like manner throughout the brain. And so if you put corrupted alpha-synuclein that's misfolded into the mouse brain surgically, these mice actually develop structures in the brain that actually closely resemble what we see in human disease. And so while this is very exciting for the field, and now a lot of, I think, preclinical trials are using these mice to test potential treatments, we have to keep in mind that the cons of this approach is that it's an acute injection. Nobody with Parkinson's disease had surgery in which they got alpha-synuclein injected into their brain, right? Also, these mice don't actually have behavior defects, and there's also different outcomes depending on where you inject this into the mouse brain. And so I think this model is very good in the sense that it mimics pathology, but it doesn't mimic the whole spectrum of Parkinson's disease, not to even mention all the non-motor symptoms that are present. Next, one model that I wanted to cover, since it just appeared a week ago in the literature, in the scientific literature, and it just so happened to come from Dr. Schlossmacher's previous mentor at Harvard, where they generated a new transgenic model of Parkinsonism by mutating the gene for alpha-synuclein and then putting a bunch of it into the mouse brain. So again, very artificial, to dose the mouse brain with that. But what was really remarkable in these mice is that when you put this misfolded alpha-synuclein into the mouse brain, these are snapshots from a video that they took. When you put the mouse on a pole, naturally mice are very shy and skittish and want to not be in an open area. So they'll climb down the pole really quickly. And the mouse, the 3K mouse, actually tries to climb down but falls several times. And what they found was most exciting for them is that when they gave these, that didn't work out. When they gave these mice L-DOPA, which is a current treatment for the motor movement symptomology of Parkinson's disease, these mice improved, showing that it's really their dopaminergic neurons that are suffering. And then here on the right, I hope you can appreciate, these mice actually accumulate Lewy body-like structures as well. And so these mice have a lot of the features of Parkinson's disease, which is very exciting for the field. Again, the pros is it's it's phenocopying, or has a lot of the features of classic PD. The cons is that this is not very representative of what happens in human. So no human has all these mutations and has tons of alpha-synuclein dumped into their brain. So we really need to find ways to mimic, somewhat the progression of Parkinson's, or what happens in Parkinson's disease in a more reliable manner. And the last model I wanted to talk about is the model that we've worked on in our lab that we've generated, and it's called the alpha-synuclein NLS model, for lack of better name. And what it consists of is actually taking alpha-synuclein and forcing it into the nucleus of cells. And so the reason we did this is, first of all, alpha-synuclein, or the name synuclein, when it was first discovered, about almost 30 years ago, came from two words, synapse and nucleus, because that's where it was originally found. And since then, a large amount of the field is really focused on the synaptic function of alpha-synuclein, just because probably 90% of it is in the synapse. But what we and others have found, this is work from Tiago Utero's group in Göttingen, Germany, they find that in humans with Lewy body dementia, you have accumulation of alpha-synuclein in the nucleus of their cells. And so what we decided to do is say, well, what happens when we force alpha-synuclein into the nucleus of these cells? And so now Haley's going to be working with these mouse models to see if they develop motor behavior defects, but also non-motor behavior defects as a result of the accumulation of alpha-synuclein in the nucleus. And so what does this all mean? Where are we at now? To follow the point of the previous discussion, I think we've made great strides in the field. I think we've come up with quite a few models. I've highlighted a few of them, one of which is the alpha-synuclein GBA interaction that Dr. Schlossmacher's lab has done some nice work on, the Parkin mutator work that Richard and Dr. Slack's lab is working on. And so there's several of these models, but there's really no coherent, unified model for Parkinson's disease. And one could argue that, A, that's because Parkinson's disease is very heterogeneous, but also what all these models really focus on is just targeting one single aspect of the disease. And so now what the field is really working on is really to try to assemble the pieces of the puzzle. So can we find a way to take each of these models, or what we like about each of these models, and integrate them together so that we can find a really nice model on which we can test preclinical treatments? And so ultimately, we'd have a better mouse model that we can test preclinical treatments. And so what I don't have time to talk to you today about, but which the bulk of my previous work in Houston, as well as some of the work in our lab does, is to identify new potential treatments. And so these all converge together that once we have a good model to test on, we can test these potential treatments and then bring them back again into the population. And so with that, I wanted to thank the Brain and Mind Research Institute, especially those who organized this. I'm sorry if I'm missing some names. And especially thank you to the Parkinson's Research Consortium that make this research possible, especially to a new researcher like me. As I'm applying for larger scale grants, it's really these grassroots efforts that make our lab able to function and to exist. And thank you to all my lab members who do all the work. Thank you. Q&A? Yeah, and then I think we have time for 5 minutes or so of questions. So for either aspects of the talk or just broad questions that you may have.
And if you'd be so kind as to just raise your hand and you get a microphone so that everybody can hear you when you have questions, okay?
You were looking at Thank you. You were working on genetic mutations, looking for new ways of attacking the disease while you were in Houston. Did that research come back to Ottawa with you, and do you have any results that you can speak to us about?
Yes, so some of the results were actually, or should be published next week, which is very exciting. What we're doing in Houston, and which I am bringing back here, although it takes some time to get things set up in the lab, is we're performing large-scale screens to disrupt the whole human genome to see what effect each of these genes have on alpha-synuclein, for example, or any other feature that you're looking for. So for in Houston, we know that alpha-synuclein is really bad, so too much is bad. That's the best way to think about alpha-synuclein. So we did a lot of screens to look for potential chemicals that could decrease alpha-synuclein levels. And so we're bringing that technology here, or I've brought it back here, and we're working with several of the members of the PRC to get that set up.
You were number 2.
Assuming that that this heterogeneous nature of Parkinson's, is there opportunities with AI to look at data from a large number of sources and use this? I understand there's someone that was with IBM that was working on some system with AI that would be helpful to Parkinson's.
So that's a very good question. Machine learning is what you're referring to, that you take the abundance of data that has been collected and you put it into a pot, into a machine, and try to come up with an answer. So this is in part what we have done and what not just our team, but also other teams have done with respect to these multiple risk factors. And one nice study that was published a year and a half ago was published in Lancet Neurology, which is one of the leading journals, that brought together geneticists and epidemiologists and clinicians. And it found in a similar approach that we took, except for adding very comprehensive genetic dissection, so to speak, from every individual, as Max mentioned. And they put this all into a machine and let the machine say, can you differentiate who has Parkinson's versus who does not? And they came up with more complicated formula, but essentially they were able to differentiate who has PD versus who doesn't in 90% of the cases. And this is also without examining a patient, but it was largely successful because it had a very detailed genetic information. Every single donor, so this is a group of people, over 800 that participated, and every single person had 500,000 pieces of DNA mapped. So a normal person can't do it. You need the machine support, but our idea is, can we potentially simplify it? And so not one approach is going to solve it all, similar to the mouse modeling, but we need to employ people who have totally different skills, not skills to study mice or work with patients, but actually mathematicians, statisticians. And so this is actually why it's really wonderful to be in this city and planning ahead for what the University of Ottawa Brain and Mind Research Institute can do in the future, is to actually bring these different components together. And so we have such a successful high-tech sector here in the city. One of the things that we want to do is reach out for them to learn about what our problems are, and for us to learn what solutions they could bring to the table. So excellent question. Sorry for that long answer.
I've been attending many conferences on Parkinson's disease, and each time I notice that the sense of smell is popping up. Is that could be a venue in order to predict Parkinson's disease?
Yeah, so you were asking whether the loss of sense of smell is helpful. Yes. So essentially every model, including ours too, has identified that the loss of sense of smell is a very strong predictor who will have Parkinson's and who will not. It is not very specific. Some of you may know that even people with Alzheimer's disease also lose their sense of smell. But if you take it together with other problems such as constipation, then it increases the chance to have Parkinson's. And we think that the reason why constipation and loss of sense of smell may both confer increased risk is it has something to do with the environment, because there's bugs, viruses, bacteria that live in our nasal cavity and in our intestinal tract. And so maybe it is a certain amount of virus or yeast or bacteria that initiate the cascade that leads to Parkinson's. So the loss of sense of smell is very helpful, but it's not the only factor that needs to be taken into account. And we're also trying to model this whether animals can lose their sense of smell, and that's also a research that's been going on here. And so this is very exciting. You need to take different clues from different directions.
Hello,
Is it on now?
can you hear me?
Yes.
I'd like you to take a broad look or tell us in a broad sense about the field of research that revolves around Parkinson's. I've read and seen blogs on the internet that seems to say this field is really is exploding, that there's lots of research going on, there's lots of trials, and surely there should be some good results coming from it. Can you tell me, as researchers in this field, whether that's true or that's hype, or how can you characterize the emerging possibilities of some really good solutions?
Yeah, I would say that the field is definitely exploding. There's a lot of great minds who are coming up with very intriguing and possible explanations for what causes Parkinson's disease, and coming up with potential treatments. I think one of the difficulties that lies in the field is to get everybody on the same page, potentially, and to begin to treat Parkinson's disease not as just a whole disorder. I think, as Dr. Schlossmacher was alluding to earlier, there are very different forms of Parkinson's disease. And I think if we start stratifying patients that way and treat each disease individually, then I think the current research that's being done would definitely benefit a lot of people.
I think, it's funny that Max, he calls me Dr. Schlossmacher, so should I call you Dr. Russo too? Please call me by my first name. He reminds me of what I said 20 years ago, that we have to get a unified perspective. And some wise owl in the audience said to me once, no, you don't want the scientists to agree on, because they haven't fixed anything in 40, 50 years. So you actually want the young people that come up with new ideas and challenge the people with the gray and white hair. So I think that the most important thing is to try to get people from different disciplines that have totally different skill sets, that are not biased, that are not dogmatic, that help us to open our eyes how things have been solved in cancer biology, in heart disease. And so my criticism would be in the field that we've been too dogmatic and too focused and too narrow in our view. And that has really changed, I think, in the last 2-3 years, that including machine learning and to incorporate different disciplines and skill sets. And that's the most exciting one, not just for us, but also for our patients, because the chance to now make a breakthrough discovery has multiplied, I think.
And actually, I would add to that point that we're learning more and more from the other fields. So cancer, especially technologically, has always had the upper hand on us for the past 50 years. I don't know if you would agree. Just because the sheer amount of patients, but mostly funding that was dumped, especially I think through the Reagan administration. And so because of that, we've kind of always been struggling behind, trying to follow what cancer has done, but I think it's always been a very good way to learn. Myself as a trainee always read the cancer papers to really understand how to tackle the problem using a different approach. So I think that's very wise.
Natasha Hollywood, who is the master organizer, just signaled 2 more questions, and then we have a few other things to get accomplished. Ma'am.
I just have a quick question. I'm curious about the alpha-synuclein. How do you get that to put it into the mice? Does that come from human beings, or is it synthetic?
That's a great question. Both. So originally, the PFF model is a synthetic, and so the synthetic model means that you can take a protein, put it in a bacteria, and force it to produce human alpha-synuclein, break open that bacteria, purify it, and so it's essentially just a drug, right? And or a compound, and you put it in the brain. But originally, what was done was post-mortem tissue that was donated was actually isolated and then put into the mouse brain. But yeah, now it's gone to the point where synthetic alpha-synuclein can actually accomplish this role. This was a big breakthrough in the field.
Yes, sir.
Does this work?
Are there definable populations which have different rates or manifestations of Parkinson's?
That's excellent. Wonderful question. Nobody really has a complete answer for it. Generally, it is thought that the rate of Parkinson's is about the same in the Western Hemisphere. But then there are some people who study populations, they're called epidemiologists, that think that maybe in India, in Africa, and even in the Northwest Territories here in Canada, the rate is lower than what would be expected. The problem is that there's what's called a selection bias, that maybe the people that are being examined are not representative of people who live in the field. Maybe we can't access all the people as a representative portion of the population. So, and are the tools the same that have been applied? By and large, there's not a huge difference as there is, for instance, for multiple sclerosis. But it may not be identical for all populations and ethnicities. We know that genetic risk varies greatly around the globe, so different populations and ethnicities have different, either higher or lower risk with respect to specific genes. So it's a good question, but the final answer is still out there. So if I may, if any of you have additional questions, maybe we can mingle afterwards. I just wanted to do two things next. The first one is we have to ask Mr. Tom Bolton to come back to the stage, because the recipient, Yolanda, was stuck in traffic and has actually arrived, and she would love to receive her diploma and her fellowship award. So Tom, thank you very much. So where's she? Oh, here. Okay, here she is. Sorry, didn't turn to the right. So, congratulations.
I was working on the experiments for this project.
Best possible answer, really. And the second thing that I'd like to do is to ask a very important person to come to the podium, Bobbie Briscoe. She is one of the, when I said earlier that a lot of the work that we've been doing was made possible by generous support from the community. Many of our community members are in the audience that supported us through various venues, the Ottawa Hospital, or the Parkinson's Research Consortium, directly to the university. So there have been many people that have supported us, and Bobbie Briscoe has played a very special role. She's one of the founding fathers of an effort that's referred to in the community as PIPER, Partners Investing in Parkinson's Research. And they have done an amazing job and have accomplished something that I thought wouldn't be possible. So to tell you a little bit about the support that funded some of the research that you've heard about, I'd like to ask Bobbie to come up and say a few words. And she has her shirt on that says PIPER. So thank you, Bobbie.
You wear the uniform. Good afternoon. It is my privilege to speak to you in my role as co-chair of PIPER this afternoon, Partners Investing in Parkinson's Research. 10 years ago, a few of my colleagues in the investment industry realized we were each impacted by Parkinson's in our lives. In my case, my husband, John. And we decided we wanted to do something meaningful and positive about this disease, and decided the best thing to do would be to invest in research. With the help and support of the Ottawa Hospital Foundation, Team PIPER was born using the Ottawa Race Weekend in May as our fundraising vehicle. The 2K walk is our main focus, although we've had some of our PIPER members do some amazing things. Mark Belfoy, who I'm not sure is here this evening, he lives with Parkinson's, and he did the half marathon on his 40th birthday. My husband and I still shoot to break the 30-minute mark on the 2K. It's an event that embraces everybody's ability to participate. Many of us love to be at the heart of the walk, while others choose to do their walk closer to home, like our PIPER members out in Canada who do their walk at the golf club, even others who support us by simply sharing their story with family and friends and asking for donations to the PIPER cause. One of our founders, Richard Shantz, has moved to British Columbia with his wife, Janet, and they continue to support PIPER from afar. It's a wonderful partnership, a partnership with our great researchers and clinicians here in Ottawa who walk with us, tour us through their labs, and report back to us on an ongoing basis with their projects. It's also a wonderful relationship that has developed as we share our challenges and exchange information with one another on how best to live with Parkinson's. I'm proud to say that this year, our 10th year, we have reached the remarkable total of $1.3 million for the extraordinary team of dedicated clinicians and researchers at the Ottawa Hospital and the University of Ottawa. And what is so meaningful to... And as Michael said earlier, every penny counts, so. What's also so meaningful to all of us is that we've been able to see the impact of the donations to our friends and family, better therapies and new treatments in the clinic. For example, we now have deep brain stimulation in Ottawa, a procedure that our dear friend Shelby, who unfortunately wasn't able to be here tonight, but she has benefited from. There are just so many advances to be proud and hopeful of. We know we are making a difference, and that inspires us to continue with our efforts. The researchers we have heard from here today need our support, and they need your support. We invite you to join us and learn more. Piper is www.piper.ca, and I'm happy to answer any questions after the presentation concludes. Thank you.
Unless Natasha signals to me that we have to add something else, I think, yes, do we have to? Okay. You should have run to the podium. Okay. This is Natasha Hollywood. She has overseen the organization and she would like to address you directly.
Actually, I just wanted to say for the awardees, because part of this evening is for them and the important part of the evening is for them. So once we're done here, if you don't mind just coming to the corner there, we'd like a group shot with Max and Michael just to help promote your achievements this evening. That's it. Thank you.
Thank you, Natasha, and I'd like to take the opportunity to thank you all for coming. It's truly an inspiration to meet and work with patients and their family members and our trainees. And I wanted to tell you where Ottawa is also leading in Canada and in North America. It's when it comes to actually approaching the disease as a community. More and more funding agencies in the United States and in Canada actually mandate that now, that whether it's Alzheimer's, Parkinson's, or cancer, that researchers not only promote their own interests and communicate their objectives, but that they actually get patients involved, patient organizations. And more around North America, patients help drive investigations, discovery, fundraising, and so the tighter the community, the higher the success rate of landing some prestigious grants for from federal support sources. So we have been living that with your help and with your support, and we're really, really grateful. I want to take the opportunity also to thank again Ms. Jensen as the president of Algonquin College for hosting us. I want to specifically thank Natasha Hollywood and her colleagues, Jennifer, Victoria, Candice, for putting this event together. It's one of 5 events this week under the Brain Health Awareness Week effort by the Brain and Mind Research Institute. I also want to acknowledge how this all began a little bit more than 5 years ago. 2 very strong supporters of the Ottawa Hospital Foundation and the university, Utra and Sam Bargava, had this idea to put this event on. It didn't start with us, it started with the community. And they said, we have to get the message out, we have to engage the community and update people as to what we're doing in different disciplines. And Sharon is smiling because she knows this is true. And we are driven not only by our own curiosity, but also by the stamina, the endurance, the persistence, and the creativity of our community members. Thank you very much. So please enjoy the rest of the evening. Thank you again for coming. I also forgot, Max did it. I didn't thank my own team to support my research activities. I'm eternally grateful. Thank you for shaming me. I had removed too many slides to make sure it all fit in. Thank you to my team, and thank you all for coming.